So one of the interesting, I think, facts about our cells is that all of them, not just neurons, actually maintain a cross membrane potential difference, like a difference in charge inside versus out. And that whole term potential difference, I want us to understand pretty well for this class. It's the idea that there is a charge difference inside versus out, but it actually derives from some of the early work. If you read the Candel chapter, for example, you see that Hodgkin and Huxley took electrodes, what they call potentials, and they took one potential, right, one electrode, and they passed it through the membrane of the giant squid axon, which was visible to the naked eye. It was a very wide diameter axon, and it's how invertebrates actually, which are not encased with a lot of the nervous system not encased in bone, right? They actually, instead of myelinating, they tend to increase the diameter of the axon of the wire, so that the current can actually move to the point of least resistance. There's a lot more space in there, so it can get away from the turbulence of the sodium and the potassium moving in and out as there's propagation, and also get away from the receptor proteins and channel proteins that stick in quite a ways into mammalian axons. So they took one potential, stuck it on the inside of the membrane. They took another potential, another electrode, and put it on the outside. And when they attached it, by a wire, there was the flow of current because there's a charge difference, and they could measure that, right, with a voltmeter, and they could put, like units on it, and you realize that there's a cross membrane, charge difference of 65 millivolts, about 65, 70. It'll vary, depending on what species we're talking about and what part of the nervous system we're talking about. But it's more negative on the inside by about 65, 70 millivolts versus the outside in a neuron at rest, right? But it turns out that most cells maintain some sort of cross membrane charge difference between those two potentials. There's a difference, but neurons are kind of unique in that they alone sort of conduct bioelectric signals, right, for long distances without any loss of signal strength. That's actually quite interesting. The way that the fidelity, the action potential is sort of maintained along the entire length without dropping off. If you, for example, think about electricity traveling in copper wires. The farther it gets from the source, right, the greater the drop in in the signal. That's why we have all those stations to boost the signal. But it's also much faster. It travels closer to the speed of light, actually, in a copper wire in terms of the movement of charged particles. While in our neurons, we're talking about in an unmyelinated neuron about 2 meters per second. With myelination, you can expand that rather dramatically, but still nowhere near the speed of light. But the consistency of that current is maintained. The action potential continues all the way without losing signal strength. That's pretty important. And the other thing is that, of course, neurons alone, possess specific intercellular between cell connections, with other nerve cells and with other tissues, including muscles, right, to move, contract muscles, and also glands, to release, chemicals, from the adrenal glands, for example. So neurons are quite unique in that way. The other thing to think about with neurons and the electrical nature of neurons is Ohm's law, right, which is voltage equals current times resistance, right? So voltage, is a measure of the charge difference between the inside and the out, is dependent upon the current, how many ions are actually flowing, right, which is dependent upon how many channels are actually open to allow for the current to flow, which is a measure of the resistance of the membrane. So V equals IR. So if you have, for example, minus 65 at rest, right, that's a charge difference. It's a certain V, and it's equal to I, the current flowing, times R, the resistance. Well, the I, the current flowing, is not very much because the voltage-gated sodium and potassium channels are closed at the resting potential of minus 65. So that's low. I is low, which means that R, the resistance, right, of the membrane, is going to be high, right? So because there are no channels that are open, it's impermeable to the passage of these ions. However, if you suddenly reduce the resistance, right, of the membrane, and we do that, of course, right, at minus 55 millivolts at the threshold potential, we open up lots and lots of sodium and potassium channels. Well, the resistance drops significantly. So what changes? Well, the current goes way up, right? And we get a big we get rapid redistribution of charges, we get big changes in the voltage, we get an action potential.